It's my pleasure this morning to introduce this section on aspects of this topic that, despite its obvious importance, is still so difficult to investigate. Our first speaker, Dr. Robert McNamara, is a professor of psychiatry and behavioral neuroscience and director of lipidomics research program at the University of Cincinnati College of Medicine. He heads a translational research program investigating the role of omega-3 polyunsaturated fatty acids in the pathophysiology, etiology, and treatment of neuropsychiatric disorders, including major depressive disorder, bipolar disorder, and ADHD.
So I'm going to try to provide a brief overview of some of the work that we're doing, some of the work that I'm currently engaged in Cincinnati, in an effort to convince you that those three things are interrelated. And I think they, by studying neural networks, which is a relatively new neuroimaging analytic technique, we can better understand how omega-3 fatty acids may contribute to the pathophysiology of major neuropsychiatric disorders, and perhaps better understand how omega-3s may actually help treat and or prevent these disorders. So one of the clues to when we try to understand the etiology of psychiatric disorders is basically retrospective data suggesting that a lot of the psychiatric disorders, their initial onset occurs during the adolescent period. And this is important for a number of reasons, and using this information to help guide us forward is the fact that this period is also associated with major neuroanatomical changes in the prefrontal cortex, particularly. And we know that the prefrontal cortex plays a very important role in regulating limbic structures, including the amygdala, which we know also plays a role in emotional processing. And that's a very dynamic developmental maturational process that occurs during this period when psychiatric disorders are initiated. Emerging evidence suggests that, neuroimaging evidence suggests that the adolescent period is also associated with rather dynamic changes in the interrelationship between the prefrontal cortex and the amygdala and how they communicate. And this ultimately is associated with how reactive the amygdala is to emotional processes. And for example, even in children that are subsyndromal, they don't have anxiety disorders, but instead exhibit elevated anxiety to demographically similar children, are exhibiting elevated functional connectivity between the prefrontal cortex and the amygdala. And we know, based on meta-analyses, now that one of the important things here, I think, is that using neuroimaging to better understand how omega-3s interact with neuropsychiatric disorders. And one of the reasons I think that is going to be important is because we now, after 20 years, have developed a relative consensus as to what specific disorders are associated with from a neuroimaging perspective. But what has emerged from this 20 years of neuroimaging research is that the prefrontal cortex to the amygdala, there's hyperactivity, hyperconnectivity, resulting in amygdala hyperreactivity to various emotional processes. And this, to me, suggests that perhaps the maturation of this process that occurs during peri-adolescent development may represent a therapeutic target in which we can intervene, particularly in at-risk kids, to help put trajectories back on a normal course. But how do we do that? What are we going to intervene with? Well, you may have guessed, one of the interesting things that has also emerged in parallel with this research is the fact that all of the major neuropsychiatric illnesses, based on meta-analyses, exhibit lower blood levels of DHA, in some cases EPA, which I think is very important. Again, many of these disorders emerge during peri-adolescent development. So we have that association. And this is just one study that we published a few years back demonstrating that the EPA plus DHA levels in the red blood cells of individuals with BP, bipolar disorder, are significantly lower than healthy controls, and those that blood levels in individuals that are at ultra-high risk for developing bipolar disorder are also significantly lower. And those at high risk, i.e. they have a family member, a biological parent with bipolar disorder, which increases their risk of developing bipolar disorder, is somewhere in between. And these levels across all these subjects are associated with both manic and depressive symptoms. So moving to the brain now, we know from our studies as well as other studies that during the peri-adolescent period, prefrontal cortex DHA levels also exhibit a linear increase, suggesting, again, that DHA or deficiency may contribute to some of these abnormal neurophysiological processes. And importantly, we find repeatedly that red blood cell DHA levels are highly correlated with rat brain DHA levels. And so what mediates interconnectivity between brain regions as well as within brain regions are synapses, as you may have guessed. And there's now a plethora of very interesting preclinical data indicating that developmental omega-3 deficiency results in a deficiency in synaptic plasticity within a number of different regions, including the hippocampus, and a significant reduction in synaptic number. And we also know that DHA dose-dependently increases synaptic dendritic spine density in animals and increases resilience to things that reduce synaptic density, including inflammatory processes. Interestingly, also, there's some evidence suggesting that DHA insufficiency also reduces synaptic pruning during normal neurodevelopmental processes in the developing visual system. And this is relevant because we know, at least in the rat, as I suggested in the intro there, as also occurs in adolescent humans, this period is associated with very robust synaptic pruning in the connections between the prefrontal cortex and the amygdala. Therefore, synaptic pruning and DHA's effects on synaptic pruning may have a significant impact on these developmental trajectories. So this leads to the hypothesis that developmental omega-3 insufficiency disrupts cortical limbic network maturation, which, as I suggested, is an endophenotype associated with psychiatric illness. And so this is a very recent study that we haven't published yet, but it's very interesting to us because it supports the proposition that omega-3 insufficiency is associated with significant reductions. If you look at the first rat brain, you can see the difference between the control animal and the omega-3 deficient animal, and this is a perinatal deficiency paradigm. And importantly, postnatal fish oil supplementation to the deficient animals is sufficient to completely restore functional connectivity within the prefrontal cortex. as well as restored prefrontal DHA levels. We also have some behavioral data suggesting that the circuit, prefrontal cortex to amygdala, is also disrupted in animals that are deficient through perinatal development, specifically impaired fear extinction, which is obviously relevant to anxiety disorders and PTSD. And also using rat neuroimaging with a 7 Tesla, we demonstrate that the functional connectivity, this is resting state, is significantly reduced in animals that have undergone omega-3 deficient diets over peri-adolescent development, suggesting that we have some kind of target engagement here. Previous studies have also demonstrated in non-human primates that there's significant changes that occur between the prefrontal cortex and temporal cortical regions as a consequence of omega-3 deficiency. We've previously demonstrated in kids with high versus low omega-3 levels in their blood, significant differences in functional connectivity between prefrontal cortex and temporal cortex. And this is a paper that's relatively old now, but we demonstrated that at least at lower statistical thresholding, prefrontal cortex activation increases substantially in response to an attention task, but corresponding reductions in temporal cortex activation are certainly consistent with increased inhibitory feedback and cortical limbic connectivity. So these data suggest that indeed, or support the proposition at least, that omega-3 supplementation will increase functional connectivity in cortical limbic networks in youth with depression. And so this is a study that we just completed and has just been accepted for publication, where we had high-risk kids, that is, they have a biological parent with bipolar disorder, substantially increasing their risk of developing bipolar disorder themselves, or at least a mood disorder, but also depressive symptoms, which again, we know from other data significantly increases their probability of developing a bipolar disorder. So this is a very interesting cohort to work with, particularly when we're investigating potential preventative interventions. And although we didn't see robust differences between our placebo in terms of depressive symptoms, we did see significant changes in global functioning measures as well as clinical ratings of global symptom improvement, CGI, which may in fact be a more sensitive measure to the positive effects of fish oil on depressive symptoms. But we did find also that significant changes in the fish oil group in the prefrontal cortex to the temporal cortex functional connectivity, and this is during visualization of emotionally negative pictures. And simultaneously, we have also seen significant reductions between the amygdala connectivity with the temporal cortex, which is sort of a top-up reduction, which we view as potentially a positive thing. And this is using graph theory technique, showing, similar to what I've shown you in the rats earlier, that the subjects receiving fish oil supplementation show significant increases in prefrontal cortex regions, as you can see here, including connectivity with the amygdala. So that went much faster than I thought it would. So perhaps this as well. So neuropsychiatric disorders, so these are my conclusions. Psychiatric disorders are associated with omega-3 deficits and abnormalities in cortical limbic functional connectivity. Developmental omega-3 fatty acids is required for optimal synaptic maturation and plasticity in rats. Developmental omega-3 insufficiency alters cortical limbic functional connectivity in rats, non-human primates, and healthy children. Omega-3 supplementation increases cortical limbic functional connectivity in adolescents with depression. So this leads to, this is obviously not conclusive, but it gives us clues into sort of how omega-3s may contribute to, or maybe sort of more causal relationships between the deficiency that we see in kids with omega-3 deficits and neurological processes which we know have been implicated in psychiatric illness. And ultimately then restoring functional connectivity or certainly normal developmental trajectories in these particular networks may contribute to the therapeutic effects of supplementation. Questions? Thank you.
Our last speaker in this section, Dr. Philippe Arvert, teaches at the University of Grenoble and the University of Inter-Ages de Dauphine. He is also on the board of the directors of the French Society of Topicology and a member of the scientific committee of SOS Addictions and Observatory of Adolescent Risk Behaviors. Dr. Arvert has spent more than 30 years on risk factors linked to addictive behaviors, particularly among adults, among young adults. And he has published numerous papers and book chapters on this subject.
Thank you. Hi, everyone. I know it's difficult to be the last speaker before lunch. I try to be not too boring. So, why today here to speak about omega-3 and aggression among healthy adults? Some of my colleagues in the past conducted a survey about aggressive behavior and extra pharmacological effect on alcohol, and we published a paper in 2009, in Journal of Experimental and Social Psychology. More people thought they drank alcohol, more they had aggressive behavior. We observed some alcohol expectancies, even when people didn't drink any alcohol. And 2 years after, some of my colleagues won IG Nobel Prize. You know, it's a prize for publication with no scientific substratum. And they won that prize because they observed, more you drink, more you think you are attractive to others. So, well, with 1 year after, Aps Zalberg, it's a psychologist from the Netherlands. He conducted a survey in jail among prisoners. And he gave them some fish oil, vitamins, and he observed, with a control group, he observed a decreasing aggressiveness in jail. So we decided to replicate that study, and at first it should have been among alcohol use disorder outpatient, but it was difficult to include enough patients, so we decided to expand the survey to general population. So here we go. As you know, omega-3 I concentrated in the brain, and my colleagues in the 2 prior presentations are on that topic. Donc, importance of fats, of omega-3 for the brain development, maturation, in terms of structure and also function. 20% of the dry weight of the brain in the nervous circuits, okay? And importance, so in the membrane elasticity. And In our neurons, there is a myelin sheath, and fats are present. Omega-3 and psychic well-being, yeah. You have many studies about the important and the presentation by my psychiatrist colleagues for people suffering from ADHD, autism, and bipolar disorder, the importance of omega-3. And this research focuses on the benefits of omega-3 fatty acid on reducing aggressive behavior. Yeah, here we go. Yeah, it was a study about the importance of PAF in autism in maybe in 2017. Meta-analysis and some other studies have shown correlation between omega-3 blood level and violence. And another ecological study in 36 countries found a negative correlation between seafood consumption and homicide rates. And so on and so on. I go to the last part of this slide with our colleague in Zalberg, and he found a 34% decrease in antisocial behavior in young male offenders. So, as I told you, we decided to replicate that study. Yeah. It was an experimental design to test the effect of omega-3 supplements of self-reported aggression. Almost 200 French adults using the local press advertisement and flyers. So some of them were dropped out of the study, and there was no differences between the two groups with omega-3 and the placebo group, and 194 participants. So yeah, we have all the improvement of ethics committee, and we explained to the people that they could receive one medication or a placebo, and they were addressed to one or the other group randomly. So 2 pills a day, 2 capsules with DHA, EPA, and each pill contained 1,000 milligrams, so 1 gram, and they took 2 grams a day. In the placebo group, we used the copra oil. Yeah. We used the Busberry aggression questionnaire, 29 items, but we focused only on the physical aggression, but there are some items about verbal aggression, anger, hostility. And here are the items. Once in a while, I can control the urge to strike another person, and so on. There was a robust intercorrelation between these items, using the Cronbach alpha items. So the questions were about the last month. We took in account also how much fish they had eaten, and an estimation of omega-3 fatty acid was calculated. Yes. And we used classical analysis analysis of variance, and we took into account some covariates. And the aggression score has been transformed, and we use the log transformation. Yeah, the two models. Yeah, and voila. To have a more normal Gaussian distribution, a log transformation was used. And we observed a decrease in this model, taking into account some covariates and the log transformation, a decrease of the aggressiveness score between placebo and omega-3 group. And we observe the same result in a univariate analysis of variance. So, analysis of variance of covariance, we observe this decrease, statistical difference. So, the effect size was of 0.31, so it was a good, important, notable, consequential effect size. And the reduction in aggression level caused by taking omega-3 supplements for fair roots was not trivial. So, yeah, it was only 6-week trial, maybe not enough to observe some other differences. And we had not the omega-3 index. It's important if maybe in the future we can use it at the beginning and at the end of the study. Yeah. As I told you, only a short-term effect. Maybe a long-term effect would be important to be studied. And it was a self-report measure of aggression. So it's not good enough. And in the future, it should be necessary, as I told you, to have the blood level of omega-3 fatty acid. Yeah. Yeah. Okay. Well, I think, yeah. We used the Omacor by Pierre Fabre company. It was very easy to use it. Our Dutch colleague, Alp, had some difficulties with his presentation of pills because it smelled fish, and it was very easy to know if you were in the control group or in the oil group. So, thank you for your attention.
So then we are ready for questions. So are there any questions for the speakers?
Hello. Hi. So I guess I have a question, and very good talks. I really enjoyed them. I have a question about crossing the blood-brain barrier, and I know people talk about circulating levels of omega-3s in relationship. And doing a lot of lipidomics recently, the molecule that's really, I think, been underappreciated, because most of us who are lipid biochemists left it in the aqueous phase, are the lysophospholipids. And now with the literature, that's the way that, or a major way that EPA and DHA crosses that blood-brain barrier through the MFSD2A receptor. I'm just wondering what your thoughts of how do you get across that blood-brain barrier. I'm also fascinated with the endocannabinoid possibility as well, because we see a lot of that in our lipidomics as well. But when you're giving these, are they being converted to these lysolipids? I know there's now animal studies where lysolipids are being supplemented to animals and much more effectively increasing brain DHA and EPA. So just your thoughts on crossing that blood-brain barrier.
I think I would say ask Richard Bazinet.
Yeah, I don't know.
I really don't.
Richard is around. No. Okay, would you like to say something about this?
So we use neuroimaging. We don't actually study
Okay.
the finer aspects of things. We're more interested in the effects on established neuroimaging outcomes.
Okay, we learned a lot from Richard's work about EPA and DHA to go into the brain. And actually, it's very effective, very efficient. And, but not like DHA that you can detect the transportation. EPA seems to exist only for a few seconds. So that's the reason why we want to investigate the EPA-derived molecule such as resolvin or endocannabinoid. But yes, it's very efficiently to go through BBB to go to the brains.
So for Dr. McNamara, you mentioned in your prospective study you used 2.1 grams of fish oil, but you didn't specify the breakdown of EPA, DHA, how much total that they got in that study. Do you remember?
It was 2 to 1, approximately.
2 to 1. And how much total? How many milligrams total would be? Was that 2.1 grams of EPA plus DHA?
Correct. Yes.
Okay. And so, how do you feel about like in the cardiovascular trials where they used up to 4 grams, got better benefits? Do you think is there any trend towards higher benefit with higher doses in the brain as well?
Well, I'm recalling a specific study where they looked at DHA supplementation for depression, and they observed that the lower dose was actually more efficacious. So it's not clear to me at this point whether or not more is better. There's probably an optimal level, and that may depend on your sort of baseline biostatus as well. But we haven't done a dose response analysis on our neuroimaging outcomes yet. It'd be very interesting to do that. But we did an open label study in depressed kids who are SSRI refractory, and this is with Barry Sears, and he insisted we do his 15 grams per day in a liquid. And we observed no difference between the 2 grams per day in that particular study, suggesting that it's not, it's those kind of doses I don't think are going to be desirable by patients. It's impossible to blind for, but it doesn't seem to have any substantial benefit.
You all three sort of mentioned the omega-3 index, and I wonder if you could reflect on what maybe you think a proper level, a protective level of the omega-3 index would be in psychiatric disorders.
Well, we already had this discussion, and I think it's, there's no reason to modify it. When we have looked at omega-3 index compared to healthies in cross-sectional studies, they're around 3%, very similar to cardiac patients, which is interesting. And incidentally, excess premature mortality in these patients is attributable, if not to suicide, then to cardiovascular-related diseases. So I'm thinking, if I had to guess, 6 to 8 would be desirable, and in fact, that's what we tend to achieve with our dosing.
I have one question, at least. It's pretty obvious that EPA and DHA, they are actually two different molecules, and they obviously have different effects. And I would like to ask, might we need with regards to the omega-3 index, might we need really an EPA index and a DHA index with regards to effects on the central nervous system?
Interesting question. So we in our studies, when we're looking at correlations with baseline symptom severity, for example, or central measures, we do look at EPA and DHA and EPA plus DHA separately, and have observed differential associations. But I can't really speak to that in as much as EPA is, as we discussed, is playing a role in the brain. Is it premature to do that? I'm not sure.
On the same topic, would it be fair to say that evidence points to DHA or treatment with DHA or mainly to alleviate deficiencies, while EPA might have a place in acute and subacute treatment?
Yes, I think so. I'm actually not sure what you're asking. So,
well, we always tend to talk about omega-3 fatty acids, in this box. But in this particular area, to me, it is pretty obvious that we really need to make a clear distinction between EPA and DHA, even also in our investigations, that we might need investigations on pure DHA and pure EPA.
I think that is the answer, that especially when we're looking at central measures, central outcome measures, neuroimaging, it's always been a curiosity to me. And now that we have high quality, pure EPA supplements that we can use, I think it would be fascinating to see what different effects have.
Further questions?
Dr. McNamara, thanks for your presentation. I'm just wondering if we think about taking the body of work that all three of you have talked about and, communicating it, dare I say it, in sound bites, because that is what, whether it's from an influencer perspective, retail perspective, or clinician's perspective, that's typically what they're looking for. If we look back at the heart health data we saw earlier, and where dosage matters, do you think that there is we should see a difference between adolescents and adults in terms of EPA versus DHA? Should we be giving more DHA to adolescents, whereas some of the research shows that EPA for depression in adults seems to be the most effective?
I'm not sure there's any evidence to suggest there's a need for differential treatment of adolescents versus adults at this point. I don't know. Dr. Su, do you want to comment on that?
Well, I think dosage is another important issue for personalized treatment. And we just published a RCT about ADHD to look at the effect of EPA and DHA. And we found that the dosage seems to be an issue, but it depends on the baseline omega-3 level. So we found that for those children with low omega-3 concentration, high dosage is good. It's better than low dosage. But for those kids with high omega-3 baseline, it's not good, and even worsening for some cognitive tests. So that's for ADHD. And for depression, also there are some studies showing that for example, from the United States, show that 1 gram of EPA seems to be better than 4 grams of EPA. But in Taiwan, I wonder if it is because of the baseline dietary consumption, but we found that there's a dosage effect, meaning higher dosage to create better effect. So I think it's an issue for personalized precision medicine.
Further questions?
You just mentioned about the difference between EPA and the DHA.
Yep.
So we did some research in depressed patients. We measured the brain image, functional image, and we found that the EPA can kind of differentiate DHA when they take in the emotional stimulation. By EPA increase more, activate more prefrontal cortex, which is important for depression. And we also did one genomic study to look at the difference between gene expression after EPA and DHA treatment. And we found that EPA specifically inhibit, for example, COX-2 gene expression and phospholipase A2 gene expression, but not DHA. So these are probably kind of overall genetic effect, not really specific to the brain, but there are some difference. Yeah. I put that research in my slides of the Go Exchange app, but I skipped that in the talk today. So if you are interested, you can read the slide in the app.
I may add that I think we have quite a lot of work to do to figure out the lipidokinetics of EPA and DHA. And in that context, you really have to treat them as two different entities. And we really need to know how these two substances distribute and accumulate in different tissues. I can mention that I've seen data from autoradiography with radio-labeled EPA, and the radioactivity accumulates in brain. But we do not know which species it actually that carries that radioactivity at that time. I think we are almost at the end of our time, and I guess everyone wants to go for lunch. Yes. Thank you very much.